Welcome to the first human disease models session. We have two of these because it is such a popular topic. I'm very happy this morning to welcome my colleague Freda Miller from the Hospital for Sick Kids as our plenary session speaker. Freda is a senior scientist in the Neurosciences and Mental Health program, a professor in molecular genetics at the University of Toronto. She holds a Canada Research Chair in developmental neurobiology. She's an international research scholar of the Howard Hughes Medical Institute and a fellow of the Royal Society of Canada and the American Association for the Advancement of Science. She has many awards and honors, and I'm very much looking forward to her talk. So we're very happy to welcome Freda this morning.
So I'd really like to thank Monica for giving me a chance to present to you some of our recent data. Now, one of the key interests of my laboratory is how during development you can go from a thin sheet of proliferating neuroepithelial precursor cells of various types and build a brain that allows me to stand up here and give you this talk today. And in that regard, what I'm going to tell you about today is the work that we've been doing asking whether we can model neurodevelopmental disorders of humans in mice, and at the end, I'm going to tell you how that work has unexpectedly led to a potential therapeutic outcome. Now, the model system that my laboratory has chosen to focus on to ask some of these questions is the developing mammalian cerebral cortex, and there are many reasons for that choice. It's the seat of many of our higher cognitive functions, and there is ample evidence in humans that have neurocognitive disorders developmentally, that there is disruption of the cerebral cortex anatomically. But to be honest, as a somewhat reductionist developmental biologist, the real reason we study the cortex of the mouse, which is our model system, not the choral cortex I was showing you before, is because the genesis of the cell types that build our brains, the neurons and the glial cells, occur in a highly stereotypic fashion in both time and space in the developing murine cortex. So if you look, oops, I knew this was going to happen. If you look during sort of mid-gestation in the mouse embryo, around about E11 to E17 or so, those precursor cells that build the cortex only make neurons. And then they switch by mechanisms we're only now starting to understand to making the glial cells of the nervous system, the astrocytes and the oligodendrocytes. So this makes this an experimentally amenable system very attractive. Now, I need you to remember two things about this system to understand this talk. The first thing that I need you to remember is that most of what I'm going to talk about to you today occurs during the period of cortical neurogenesis. So that would be this time frame between about embryonic day 12 to embryonic day 17 in a mouse. And at that time, the two predominant cell types that are in the cortex are the neural stem cells, or neural precursors, I'll probably be calling them, these radial precursor cells down here, as well as the newborn neuronal progeny that they generate. The second thing I need you to know to understand this talk is that these cortical precursors build the cortex, but the other thing that they do is they populate, in part, the adult neural stem cell pools in our brain. And in fact, there's a subset of precursor cells living in here that would actually fulfill the criteria for bona fide stem cells, since they persist into adulthood and they then live in the forebrain neural stem cell niche, where they play an important role generating different cell types in the adult brain. So we became interested quite a while ago now in the idea that human cognitive disorders, especially those that occur developmentally, may arise in part by perturbing development of these relatively early neural precursor cell populations in the embryonic brain, including in the cortex. And with that in mind, we did a whole set of studies, which I'm just going to briefly touch on today because of time constraints, where we actually looked at genes that were associated with cognitive dysfunction. And most recently, the genes we've been looking at are those that have been associated with autism spectrum disorder, or ASD. And in that regard, I just want to give you two conclusions that came of that work. work. First of all is that many of the genes associated with these neurodevelopmental disorders, even though most people focus on neurons, do play a really important role in regulating, for example, these cortical precursors in the period of neurogenesis. The second take-home message was that when you perturb these genes in mice, the way you would perturb them in humans, in these cortical precursor cells, that this in fact does lead to detriments in the way the brain functions, the way it's built, and even ultimately, in some cases at least, in behavioral deficits. Well, I'm not going to talk about these genes in any detail at all. Some of our more recent ASD work is here. But I want to introduce you to the fact that that encouraged us then to take the next step and to try to ask whether in a mouse, by studying this developing cortical system, we can model the kinds of gene-environment interactions that all of us in this room probably believe are important for establishment of these genetic disorders as cognitive disorders in humans. And the particular maternal, I'm going to talk about maternal environment, and the particular influence I'm going to talk about today is maternal diabetes. And this is work that really started with a fantastic postdoc in the lab, Guangyang, who's co-supervised by David Kaplan. And it arose as a consequence of a number of really quite lovely studies that have been published over the past few years, showing an epidemiological association between maternal diabetes and autism spectrum disorder in children. Moreover, what really piqued our interest in this was that these studies showed that there is an association in humans if those mothers are diagnosed with maternal diabetes at or before 26 weeks of gestation. And this, in humans, the period of peak cortical neurogenesis is about 15 weeks to about 22 weeks. So what this told us is that this period of vulnerability actually corresponded to the period that we studied during mouse development. So what might we study about maternal diabetes that would actually give us insights into this gene-environment interaction? Well, I need to introduce you to a metabolic pathway. And it's a simple one, so it won't be too hard to get your head around it, but there's two reasons you want to know about this pathway. Let me talk about it first. Normally, all of us, we all have a lot of glycolysis going on, and under normal conditions in normal individuals, there is production of this toxic intermediate metabolite called methylglyoxal, but methylglyoxal is very quickly detoxified by an enzyme system called the glyoxalase system, which includes a very important enzyme called glyoxalase 1, or GLO1, as I will likely call it for the rest of the talk. Now, however, in diabetes, including maternal diabetes, you get aberrant overproduction of this methylglyoxal because it can't be detoxified quickly enough. Unfortunately, methylglyoxal gets in and out of cells very readily, so what that means is that people with diabetes, including maternal diabetes, have much higher levels of circulating methylglyoxal, which then can go back into other cells again. So this can become a very systemic effect. With specific regard to maternal diabetes, it's been shown that newborns who have mothers who are diabetic actually themselves have higher levels of circulating methylglyoxal, arguing that this really does get everywhere. Well, that's great, but the reason we became really interested in this is because this enzyme here, glyoxalase 1 or GLO1, there are actually a number of studies in the literature indicating that GLO1 is mutated in subsets of individuals who have different kinds of neurocognitive disorders. And it's mutated in such a way that the enzyme has decreased activity. So now we have two ways you could get this increased methylglyoxal floating around. One is by aberrant glycolysis in things like diabetes, and the second is by genetic mutations in this GLO1 enzyme. So Huang thought, well, this might be a really good example where you could have a genetic predisposition and an environmental influence actually impacting on the same cellular substrate, if you will. So with this in mind, Guang went to test this idea that not only might this happen, but it might be detrimental for cortical development. And the first thing that he did is simply to knock down GLO1 in an effort to model the kinds of things that might happen in these humans that have these loss of function mutations in the GLO1 gene. And the way he did that took advantage of the fact that these cortical precursors that we study live immediately adjacent to the lateral ventricle in the embryo. So what that means is that you can actually take these embryos out with still in their sacs. You can, at about embryonic day 13 or 14, we do this. You can visualize these lateral ventricles and you can inject things into them. Now, if you inject in things like DNA, then what you can do is just put some little paddles across the head of the embryo, and then you can give a little electric jolt, and what that does is it drives the DNA into those cortical precursors specifically, and then we can put them back into their mothers and let them go as long as you like. So this is the experiment that Guang did. He used this in utero electroporation technique to put in shRNAs to GLO1, specifically into these cortical precursors, and asked what happened. And the first thing he asked was, were there long-term consequences of knocking down GLO1 in these cortical precursors during embryogenesis? And the partial answer to that question is shown on this slide. What you're looking at here, just to explain the experiment a bit better, electroporation with GLO1 shRNAs at embryonic day 13, and analysis postnatally at postnatal day 4. Now, I need to tell you that when we put in these shRNAs, we usually put in a reporter gene at the same time, like EGFP, so you can visualize the transfected cells and their progeny very easily. So if you look at this left panel here, the left image, this is the cortex of a mouse that has been electroporated with a control shRNA. This is the bottom of the cortex right there, the top is up here, and all of these are cortical neurons, and what you can see is that the green cells are mostly in the top part of the cortex. And I can tell you these other colors are simply different neuronal phenotypes. Now, this is the one that's been electroporated with the GLO1 shRNA, and it doesn't take a rocket scientist to say, oh, the green cells are completely messed up and where they're localized. And as a matter of fact, you can go and quantify that. Moreover, you can show that in data I'm just going to tell you about, that this disruption of localization of these cells coincides to a perturbation in neuronal subtype specification. So yes, GLO1 knockdown, as might occur in human beings, is not good for development of the cortex. Well, why does this happen? Well, in order to ask this question, Guang did the very same experiment, except now he looked much more acutely, perhaps 2 or 3 days following the GLO1 knockdown, and asked what happened. I'm showing you that on this slide here. This is electroporation at E13, analysis 3 days later at embryonic day 16. Again, the pictures are very similar. This is the embryonic cortex. This is a control shRNA we've put in here, EGFP to follow the transfected cells. And the bottom of the cortex is at the bottom here, and the top of the cortex is here. All of the precursor cells live here, so that's what all these green cells are. And all these cells that are migrating up and away are newborn neurons. This is what the control looks like, and this is what the GLO1 shRNA looks like. And again, you can see there's a lot of cells in the wrong place. Moreover, I can tell you that if you go on and actually do co-labeling with phenotype-specific markers, that what you see is that there's a relatively robust depletion of these cortical precursor cells. So the white bar would be control shRNA, and the green is the GLO1 knockdown. And you can see that even within several days, you get there's only half as many precursors left. I can tell you that the reason you get this depletion of these precursors is because there's this GLO1 knockdown causes very rapid delamination of the precursors away from the apical epithelium that they normally live in. That leads to aberrant and abrupt neurogenesis and genesis of neurons. And as I've already told you, the long-term consequences of that are disrupted cortical development. Well, I've told you one half of the story. I've told you, okay, this is where we're trying to model the genetic perturbation in humans, that is this low decreased enzyme activity. Well, what about the other part of the story? What about the environmental influence? Well, I've already told you that we suspect, that we know actually during maternal diabetes, that the embryo is being exposed to more of this circulating methylglyoxal from the mother. So the question then becomes, can we model that in a mouse? And in order to do that, we took a very straightforward approach. Guang simply took these pregnant mother mice at about gestational day 13, 14. He injected them with methylglyoxal once a day till the mice were born. And then we simply analyzed the brains of those embryos, either as embryos or as a postnatal offspring. And what I'm going to tell you to understand the next slide is that at the same time he started doing these methylglyoxal injections, he also injected those mothers with BRDU so that he could actually see dividing cells and all the progeny of those dividing cells. So what did he see? Well, this is becoming familiar to you now. The cortex, this is with 3 days of these methylglyoxal injections, so at about embryonic day 16, 17. Bottom of the cortex, top of the cortex. In this case, we've analyzed BRDU. These are the precursor zones down here. This is an embryo from a mother injected with PBS, and this is an embryo from a mother injected with methylglyoxal, and I think you can see just by looking at the precursor zone that there are fewer red cells here than here. And as a matter of fact, if you go in and quantify that better with cell type specific markers, that's indeed what you see. That just as what we saw with GLO1 knockdown, that this increased circulating maternal methylglyoxal is sufficient to deplete this cortical precursor pool, with the white being mothers that are PBS injected and the green methylglyoxal injected. Well, does then this exposure to a maternal environment with increased circulating methylglyoxal also have long-term consequences for the brain? And in order to ask that, we now turned our attention to these neural stem cells that come from a subpopulation of these cortical precursors during embryogenesis. And these live in the front of your brain in the subventricular zone niche. And so we simply went in and quantified them. And now the result was that indeed there was a long-term depletion even of these adult neural stem cells as a consequence of transient embryonic exposure to increased circulating maternal methylglyoxal. And that's shown here. The white bars are adult offspring of mothers injected with PBS, and here's adult offspring of mothers injected with methylglyoxal. Now, I'm going to tell you and not show you again because of time constraints that we then went on to ask whether this kind of a change, a long-term change, was accompanied by any long-term changes in behavior. And what I will tell you is we collaborated with Paul Franklin, another one of our colleagues at Sick Kids, who's an expert in these kind of behavioral analyses. And we showed that indeed this transient exposure to maternal methylglyoxal was sufficient to lead to aberrant social type behaviors that some of us think might, we at least hope, are reminiscent of something that happens in human children with ASD. So now what I've told you is that basically there are two ways you can impinge upon this system detrimentally with the same net outcomes. Acute effects on embryonic cortical development, which then cause long-term aberrations both in the adult or the postnatal cortex as well as adult neural stem cell pools. But I have only inferred that this happens in maternal diabetes because I've been telling you that you have increased circulating methylglyoxal. Well, could we really ask whether this might happen in a maternal diabetes model? Well, in order to do that, we took advantage of the fact that, as many people in this room will know, that there's leptin receptor mice that are either heterozygous or knocked out. And what I can tell you is that females that are heterozygous for the leptin receptor, under normal circumstances, have pretty good glucose metabolism. But once they get pregnant, actually the glucose metabolism goes awry and they have a type of maternal diabetes. So now this sets you up to actually model what might happen in humans. You can take these leptin receptor het mice, females, cross them to wild type males, and then you can study the wild type offspring of those matings that are exposed to a maternal diabetes environment but are completely wild type in terms of their own genomes. And that's the experiment we did. Again, initially we simply injected BRDU around about gestational day 13, 14. And what I'm going to show you here is that just as we had seen when we injected an increased circulating methylglyoxal, that indeed we had a depletion acutely of these cortical precursor cells. And here is the wild type offspring of wild type mothers, and here is the wild type offspring of these leptin heterozygote maternal diabetes mothers. What about long-term consequences? Well, again, one of the things that we measured was these adult neural stem cell pools, the same pools that we had measured with increased circulating methylglyoxal, and here are the results. And again, just as we had seen with increased circulating maternal methylglyoxal, this transient acute perturbation and depletion of these cortical precursors led to a long-term a decrease in the numbers of adult neural stem cells in the brains of these mice. This is the controls. These are wild-type offspring of wild-type mothers, and these are wild-type adult offspring of these maternal diabetes mice. So, what I've told you then is that there are two ways you can impinge on this pathway adversely, one genetic, one a maternal environmental perturbation, and that together those perturbations can lead to enhanced circulation of a toxic metabolite, which can perturb cortical neurogenesis and have long-term effects not just on the structure of the cortex, but also on things like adult neural stem cell pools and ultimately behavior. But I think the really important thing about this study isn't necessarily the specifics of the metabolic pathway we're impinging upon here, but I think that those of us who are interested in neurodevelopmental disorders like ASD, where we know that there are hundreds of genes associated with it, and we know that something environmental is heavily determining the penetrance of those genes, that this provides a model for us to start thinking about the ways that genetic predisposition and an adverse environment could perhaps collaborate together in an adverse way to lead to increased penetrance of a neurodevelopmental aberrant phenotype. So one of the things that people in my labs say to me when I talk about stuff like this is they say, oh, well, that's really interesting scientifically, but you know what? It's kind of depressing because what you're telling us is that actually some of the things that go wrong are happening pretty early during embryogenesis. So what I'd like to do is rather than end on that note, I'd like to end on a positive note and tell you a story that started very much with a similar starting point but came to a potential therapeutic outcome. And I'm going to kind of blow through this because it's all been published mostly, but I just wanted you to have a message that there is serendipitous hope in these kinds of studies. Now, I told you we were studying genes that are associated with human aberrant neurodevelopmental disorders. And Jing Wang, who was a postdoc in my lab at the time, who now has her own lab at University of Ottawa, focused on one gene, which encodes a very well-characterized proteins, Crab binding protein, or CBP. And the reason she focused on it is because when humans have a mutant allele of CBP and are haploinsufficient, this leads to something called Rubinstein-Taybi syndrome, where these kids unfortunately have quite severe intellectual disability. So Jing did kind of what I've been telling you about. She asked whether CBP was important in these neural precursors, and if when you disrupted it, it led to aberrant things. And she did a really nice, very basic science kind of oriented project. She found that in fact environmental influences can turn on atypical protein kinase C, and this will lead to phosphorylation of CBP, and then CBP comes down onto the promoters of genes that are important for genesis of neurons and glia. It'll lead to acetylation of histones locally, and that whole process will push these neural precursor cells to differentiate into either neurons or glia. It's kind of just a generic pro-differentiation switch. Well, I wouldn't be telling you this story except that at the time we were doing that work, our collaborator on this paper, Fred Wandesford at Johns Hopkins, published a paper in Cell showing that a very widely used and safe type 2 diabetes drug, Metformin, actually can turn on this very same pathway in liver cells and in so doing regulate gluconeogenesis. Well, this became of great interest to us because Metformin crosses the blood-brain barrier and has been used safely in individuals from 5 to 105. And this is against a background of many people in the stem cell community wondering whether we could actually pharmacologically recruit endogenous tissue stem cells to, for example, promote tissue repair. So with this in mind, Jing then went on and collaborated again with Paul Franklin and asked whether or not Metformin could recruit these endogenous adult neural stem cells that are in all of our brains. And basically, we just gave generic wild-type adult mice Metformin every day for a variety of periods. I think in this particular experiment, it was for 3 weeks. And we asked whether we saw increased differentiation of adult neural stem cells, in this case in the hippocampus. And the results of that are shown in this down here. These are the siblings, the adult siblings that were just given PBS injections, and now here's the adult siblings that were given Metformin. This is the number of new neurons generated over this period. And you can see this is a pretty good increase. Paul then went on in a series of studies that I'm not going to go into detail about to show that this increased neurogenesis in these adult mice actually enhanced cognitive performance on a type of flexibility of spatial learning task that is thought to require these new neurons. Well, as I always joke in the lab, we didn't go into this to make smart drugs for university students. We went into this with the idea you could perhaps promote brain repair, albeit perhaps not in a big way, but some. And this is the point at which another colleague of mine, Cindi Morsheads, who is a really dear friend and collaborator, also at University of Toronto, came into the story. And this is Cindy's work. Her lab did this study, not I. She asked whether metformin could promote brain repair. Cindi used a young mouse model with the idea that children and young mice have many more neural precursor cells in their brains than do the rest of us sitting here right now. And so if you were going to use a therapy like this, perhaps that would be the optimal population. And she basically gave them this little hypoxia-ischemic lesion. And then a day later, she gave them metformin for a week. And then a week after that, she tested them functionally. I'm just going to walk you through this functional test very quickly. The test was simply, you put them in a cylinder, and if they use both forepaws equally normally is what they would do as they explore, and that's what you see with this white bar under control. If now you give them this hypoxia-ischemic lesion, this kind of damages one side more than the other, so now they prefer one paw over the other, and that's indeed what you see in that center bar. But remarkably, if you give them metformin for this week, you get complete functional recovery. Cindi then went on to do a series of lineage tracing studies to show that this functional recovery was coincident with a really robust increase in genesis of both neurons, and in this case, what I'm showing you, the data from her paper, is in oligodendrocytes. So you can see these are really very large increases. So on the basis of these kinds of preclinical data and on the basis of the fact that metformin has been used very safely in children and relatively chronically as well, we're now in a pilot clinical trial. When I say we, what I really mean, even though I talk to them a lot, I'm not doing it, is my neuropsychologist clinical colleague, Dawn Mabbitt at Sick Kids, together with Eric Buffet, a neuro-oncologist, and we're asking whether in human children metformin can promote any indices of brain repair. The population of children we're using are a very special population. They're children who have acquired brain injury as a consequence of their curative treatment for brain tumors at an earlier stage of their life. Many of these children had medulloblastoma. The good news about kids with medulloblastoma is that many of them live now. The bad news is the reason they live is because we give them cranial irradiation in addition to their chemotherapy. So instead of going like this cognitively as they develop, those kids are kind of like this. So Don is giving them metformin, he's measuring cognitive outputs, and he's doing a lot of brain imaging on them to see if we can see, for example, enhanced myelination due to a whole bunch of new oligodendrocytes being made. So I just wanted to end there and tell you that I hope that I've convinced you that not only does this kind of science give us unexpected insights into how the brain is built, but it also provides us both with information about what goes wrong, as well as serendipitously, at least in this case, a potential therapeutic output. And to really emphasize that the whole diabetes study was Guangyang with some help from two others in the lab, Suraj and Ignacio. All of them are co-supervised by David. Jing, really, I've got to give it to her for the metformin studies, and some really wonderful collaborators in Toronto. So thank you.
Any questions, Freda? I have one. Have you considered the use of metformin in other neurodevelopmental disorders?
Yeah, that's an excellent question, and you know it's a really obvious one because it has really been safely used for metabolic conditions in children from about age of 5 on, and so actually Dawn is the person who is really spearheading that. Because of the oligodendrocyte thing, we're looking at kids who have types of demyelinating disorders as a next target, and we've been doing the preclinical work as a group to ask that. And the other group that Dawn is really interested in, that particular neonatal hypoxia-ischemia model is thought to be kind of a model either of pediatric stroke or of cerebral palsy. And so there's a big group of sick kids, just part of this big neurodevelopmental disorder thing, who got funding to potentially go to a cerebral palsy trial. That's going to be hard because the reason we picked these kids that were treated for brain tumors is because they're a relatively homogeneous population without ongoing problems that we know about. It was an acute injury, basically. And so I think that it's going to be easier to get significance clinically out of any of your outcome measures.
Very good. No other questions? Well, that
Great.
was a wonderful talk. Thank you so
Thank you,
much,
Monica.
Freda.